Welcome to the webinar today, HEPATOPAC, a long-term hepatocyte co-culture model for ADME tox applications. This is the first in a series of 5 webinars in which we'll be talking about novel applications for HEPATOPAC. Hi, everybody. I'm Mark Warren. I'll be facilitating the webinar today. I'm Senior Director of Operations at our Santa Clara office for BioAVT, where I oversee our team of study directors and assay scientists who run transporter assays as well as HEPATOPAC studies. I work with clients to help design and implement drug transporter research studies that enable them to achieve their research goals. So before I introduce our speaker today, Dr. Deepan Vyas, I'd like to take just a couple of minutes to tell you about BioIVT. So BioIVT was founded back in 1981, and we have grown to become the world's largest provider of biospecimens and research services to the pharmaceutical and diagnostics industry. We are dedicated to providing the biospecimens and services that our customers need in order to help them advance their discovery, preclinical, and drug development programs. We have grown both organically and through strategic acquisitions that have enabled us to develop a comprehensive product portfolio and become the preferred provider for global biopharmaceutical clients. Because I'm a legacy Optivia person, I'll highlight that Optivia joined the BioIVT family back in 2018, which brought drug transporter research services into the BioIVT portfolio. Deepan, our speaker today, originally joined the legacy Qualis lab in North Carolina and worked with technologies like CDLE and BClear, which is used to estimate hepatobiliary disposition. Our 2018 acquisition of Ascendance brought the HEPATOPAC technology into BioIVT, further expanding our offerings. Other acquisitions like those of Cypex and Xenotech in 2022 have greatly expanded our ADME tox services and products. We have invested in sourcing products globally and in extensive global distribution. We stage the products in regions where our customers are located, which helps ensure short turnaround times and enables us to get products into our customers' hands when they need them. Underpinning everything we do is our commitment to ethical sourcing, quality, and a sincere desire to work as your partner, a partner to the research community. BioIVT's portfolio, here's a snapshot of that portfolio, which includes the 4 different areas: products that are used in normal and gene therapy programs, normal and C-state biospecimens, and products and services that are used to evaluate the ADME properties of drugs. Our ADME group has the industry's largest portfolio of hepatocytes, subcellular fractions, and recombinant enzymes, as well as other products to support ADME tox and DMPK research. Our team of experts routinely works with clients to help them identify the hepatocyte lots having the specific requirements needed to meet their specific requirements. Our ADME services group provides individual studies as well as integrated programs based on our clients' needs. Many of our clients actually start with a consulting engagement in which we provide expert advice to help design ADME programs, things like assisting with gap analysis, assessing existing data packages, or helping them prepare for regulatory meetings. We offer full in vitro ADME programs in our labs in Kansas City, Santa Clara, and Durham, North Carolina. Personally, I focus on drug transporter research, and I have led thousands of studies investigating the role transporters have in drug pharmacokinetics. I'd be really happy to talk to you more about drug transporters, but that's going to have to wait for an additional time. We have partnerships that enable us to integrate in vivo studies into preclinical programs. We also have extensive capabilities for bioanalysis to support integrated programs or provide as standalone services. Please visit us at bioavt.com to learn more about our product portfolio and our services. So with no further ado, it's my pleasure to introduce my colleague, Dr. Deepan Vyas. Dr. Vyas, he's a study director in the ADME services group here at BioIVT. In this role, he helps develop programs to address pharmacokinetic questions that may not always be sufficiently answered using conventional in vitro hepatocyte models. He advises clients on the use of BioIVT's proprietary BClear, CDLE, and HEPATOPAC products and designs studies using these technologies in our lab. Before he joined us at BioIVT, Dr. Vyas led the development of multicellular liver organize, an in vitro platform for drug testing and disease research at BioOrg. he obtained his PhD in integrative physiology and pharmacology from Wake Forest University. His doctoral training there was focused on the development of tissue-engineered liver organoids using stem cells for disease research and organ transplant programs. He did this at the Wake Forest Institute for Regenerative Medicine. Deepan, we really look forward to hearing your recommendations when to use HEPATOPAC for ADME tox programs. And I will turn it over to you at this point.
All right. Mark, first of all, thank you for a very kind introduction. Good morning, everyone. My name is Deepan Vyas, and today I am going to talk about HEPATOPAC. But first of all, I would like to thank everyone for taking time to join this webinar, the first webinar of our HEPATOPAC webinar series. Today, I will provide an overview of HEPATOPAC and review several talks and ADME applications where HEPATOPAC can provide valuable insight. What is HEPATOPAC? The HEPATOPAC model is a bioengineered in vitro co-culture of primary hepatocytes and stromal cells, commonly known as fibroblast. Using microfabrication tools, islands with collagen are created for attachment of hepatocytes. And following hepatocyte attachment, stromal cells are added and the cultures are matured to form a co-culture. Precise arrangement of hepatocytes and stromal cells using the microfabrication allows preparation of stable and reproducible cultures that are highly functional for longer terms over 4 weeks, and also secreting high levels of liver-specific proteins, albumin and urea, compared to random co-cultures. The hepatocyte islands in HEPATOPAC show morphological features similar to standard hepatocyte monoculture, but sustained over longer culture durations. The presence of robust bile canaliculi after 14 days of culture and canalicular transport proteins expression leads to higher activity levels of uptake and efflux transporters, which enables accurate drug disposition, safety, and efficacy assessment. For drug metabolism research using in vitro models, it is critical to have a sustained functional activity of drug metabolizing enzymes. HEPATOPAC are capable in maintaining high levels of phase 1 and phase 2 enzymes over 4 weeks of culture, as demonstrated in this study here, performed using 3 different donors. You can see the activity of major CYP enzymes, CYP3A4, CYP2D6, 1A2, 2B6, and the phase 2 enzymes sustained over 3 weeks or over 4 weeks in this case. How does HEPATOPAC compare to other in vitro models? Roche performed a study to compare in vitro models such as HEPARG, HUREL, and HEPATOPAC considered long-term in vitro liver models and HepG2 and iPSC-derived hepatocytes, which are replacement also for standard hepatocyte cultures. And in this study, they evaluated metabolic activity across diverse set of phase 1 and phase 2 enzymes. HEPARG, HUREL, and HEPATOPAC demonstrated similar average metabolic activity compared to suspension cultures. However, in terms of mean CYP activity, HEPATOPAC model showed higher or similar CYP activity index compared at day 8 compared to primary suspension hepatocytes on day 0, while the HEPARG and HUREL had 3 to 4-fold lower CYP activity index compared to suspension culture. HEPATOPAC is available for human and preclinical species. The preclinical species available are monkey, dog, rat, and mouse. We also have various strains available for monkey, rat, and mouse species. The HEPATOPAC plate format typically is a 96 or a 24 well format. The dog's HEPATOPAC is only available in 24 well format. The 96 well HEPATOPAC plates are also available in high content imaging format that are black walled and optically clear, that allows for doing in vitro toxicity assessment using high content imaging. HEPATOPAC cultures can be are received as ready to use mature cultures, so no plating is required for end users. The culture medium and application medium suitable for the application that you're looking for is also included in the kit. Should you choose to receive HEPATOPAC shipment, it arrives in 3 different boxes. One box has HEPATOPAC plates, the other box has culture medium supplements, and the third box typically has the frozen supplements that are required for the supplementation of the application medium. Now that we have established that HEPATOPAC is a stable, long-term in vitro co-culture model with high functionality, we will review the role of HEPATOPAC in various ADME tox applications. The list here for the application of HEPATOPAC is not exhaustive, but over the next few weeks of webinars, we will review most of these applications. Today, we will review the metabolism and toxicity applications of HEPATOPAC, and I will walk you through some standard study designs we incorporate in BioIVT ADME tox contract research services. So when should researchers consider using HEPATOPAC cultures for metabolism and tox studies? If your compound has low rate of clearance, HEPATOPAC cultures can provide longer incubations for clearance predictions and metabolite profiling. If also long-term exposure is needed to mimic in vivo conditions and repeat dosing regimen, HEPATOPAC can be considered for toxicity evaluation. Usually, the low turnover compounds are attractive candidates for using HEPATOPAC cultures for various metabolism and toxicity applications. When do you initiate studies using HEPATOPAC? As I mentioned earlier, HEPATOPAC are plated. First of all, hepatocytes are seated on a collagen-coated islands, and then fibroblasts are seated. Once this plating happens, we have about few days, 4 or 5 days of stabilization period for the cultures. Usually, it takes about 7 days overall for co-cultures to stabilize in the function. As you can see here, for the CYP3A4 activity, the stabilization occurs around day 7 and is consistent through 3 weeks. So all compound incubations typically are initiated after stabilization period. If you think about how this process works logistically, on day 5, after HEPATOPAC cultures are matured, they are shipped, and on day 6, they are received. Once they are received, they undergo 48 hours of incubation for stabilization after the shipment. So normally, most of the studies we carry out begin on day 8. There are some exceptions to that that we will review in some other webinars later on. Accurate prediction of in vivo hepatic drug clearance using in vitro assays is important, right, to properly estimate clinical dosing regimen. The clearance of low turnover compounds is especially difficult to predict using short-lived hepatocyte suspensions, which last only 4 hours, and also functionally declining primary human hepatocyte monolayers, which last up to 4 or 5 days, but requiring multiple media changes. HEPATOPAC has been widely characterized for prediction of metabolic clearance. In this study, 15 reference compounds with low to intermediate clearance were evaluated for clearance prediction, comparing into in vivo observed clearance. HEPATOPAC showed 93% accuracy in predicting clearance within 3-fold of the observed clinical values and was superior to suspension and monolayer cultures. High correlation was also observed across donors when clearance rates of different compounds that have higher clearance rate or lower clearance rate are evaluated. In this case, you have on top, the clearance rates of the compounds are greater than 5. If the clearance rate of the compounds are lower than 5, it also still accurately predicts, and there is high correlation across the donors. We can also use HEPATOPAC for hepatic clearance prediction of clinical candidates. In this study, Roche evaluated 8 clinical candidates, and they calculated hepatic clearance using well-stirred model. The hepatic clearance accurately predicted 5 out of 8 drugs within 2-fold and 6 out of 8 drugs within 3-fold of in vivo intrinsic clearance. Now let me walk you through a standard metabolic stability study design that we incorporate at BioIVT for services projects. Typically, a stability study involves a 96-well format of HEPATOPAC cultures or 24-well format for a dog. and different donors can be used. As mentioned earlier, different species are available and different strains are available. The control compounds we use in metabolic stability studies typically is a tolbutamide, a low turnover compound. We also incorporate quinidine, which is an intermediate clearance compound, and midazolam, a high clearance compound, if requested. The metabolic stability studies in HEPATOPAC are typically done at a very low concentrations, usually around 1 micromolar. And we typically collect about 7 time points over 168 hours. The stability study is usually a single incubation, a continuous incubation for 7 days with no media changes. And the endpoints typically involve supernatants and lysates collected combined from the incubations, and the parent compound is quantified using LCMS. The system controls we incorporate in a stability study is stromal-only cultures to measure the contribution from stromal cells in the metabolism of several drugs from some phase 2 enzymes that are present in stromal cells. If we observe any parent depletion in stromal cells, we correct for it in calculating our intrinsic in vitro clearance. The acellular wells are also included, which is the compound incubated just in media over 7 days to test the chemical stability of the compounds. HEPATOPAC can clearly identify different clearance profiles of compounds within the same plate. You can see here there are two different compounds with different clearance profile, and also we have our control tolbutamide here. This is one of the deliverables of metabolic stability study, where we see the parent depletion over time. The clearance parameters are calculated, intrinsic clearance parameters are calculated using scaling factors that are available for different species. And hepatic clearance can also be calculated using this Wellsted model equation if fraction of unbound values for the compound in plasma is also available. The low turnover compounds usually are metabolized very slowly over time, generating metabolites that may not be captured using conventional suspension cultures or microsomes over 4 hours. Due to short incubation periods. So it may not correlate well with clinical metabolite profiles, the profiles generated from these standard conventional cultures. The continuous incubation that HEPATOPAC provides up to 7 days without any media change allows for generation of detectable levels of metabolites, thus providing more in vivo relevant metabolite profiles. In a study conducted by Pfizer, evaluating 27 compounds metabolite profiles, HEPATOPAC detected 75% of total major circulating metabolites and 67% of secondary major circulating metabolites. When compared with suspension hepatocytes, S9 fractions, and microsomes, it clearly outperformed. Some other studies have also investigated successful formation of human unique metabolites in human HEPATOPAC and other species that may or may not be detected in some of the in vivo preclinical species studies. Now I'll walk you through a MetID study design for low turnover compounds using HEPATOPAC. For MetID studies, we use a 24-well HEPATOPAC culture format, and the dosing concentration is typically a little bit higher than our stability studies, and usually it's 10 micromolar. We can collect up to 4 time points over 7 days, but that's flexible. If the study design requires only 2 time points to be collected, we would typically collect a 0-hour and 168-hour time point. The replicates in the MetID studies are typically 1 per time point. The endpoints, again, is supernatants and lysates collected from incubations. And LCMS analysis is done for metabolic profiling. The deliverables usually include summary of relative abundance of the metabolites, the metabolic profiling across multiple species, and metabolic structural elucidation up to 15. But if more are required, we can certainly do that as well. There are different ways to customize a MetID study. One of the ways to customize it is using multiple species on the same plate. which can help you review species differences, and I'll also identify which preclinical species are going to form the human-specific metabolites or more human-relevant metabolite profile. Now let's switch gears and talk a little bit about toxicity application of HEPATOPAC. I wanted to talk about this interesting case of fieldred, which was an antiviral nucleoside came into clinic in the 90s for Hep B virus treatment. But 7 out of 15 patients developed hepatic failure, and there were several deaths, stopping the drug from progressing into clinical trials further. The preclinical studies using fieldred in different species did not show any sign of toxicity. Even the 10-week rat study at 1000x human dose showed no ALT or AST increase. Interestingly, woodchuck was the only model that effectively captured fieldred-induced liver injury. The toxicity was due to defective and reduced amounts of mitochondria. And even the human hepatocyte sandwich cultures were not able to capture this toxicity. When HEPATOPAC model was deployed for evaluating fieldred toxicity, 5 different donors were tested. For the 5 different human HEPATOPAC donors were tested, and comparison was done with rat HEPATOPAC. What we observed was that the human HEPATOPAC donors accurately captured fieldred toxicity, and you can see that the rat species did not show any toxicity, showing that we can also see species differences in toxicity profiles that can be captured using HEPATOPAC. HEPATOPAC can also distinguish between toxic and non-toxic structural analogs. The fieldred had a stereoisomer FIRU, which differs only in orientation at 2-fluorine atom. And when tested for hepatotoxicity, this compound showed toxicity. The fieldred showed toxicity in human donors, and FIRU did not show any toxicity in human donors. And the no toxicity observed with fieldred was again captured in rat model in HEPATOPAC. Several other studies have looked at some other analogs as well, and there will be some more discussion on this in our next webinar. But you can see here that the different analogs can be tested for toxicity, and HEPATOPAC accurately captures the differences between toxic and non-toxic analogs. Let's review the toxicity assay design in HEPATOPAC. The HEPATOPAC allows for repeated dosing at physiologically relevant doses. The studies can be customized in a lot of different ways, but a standard study design, let's say for our services project, involves 4 doses of 8 concentrations of a compound over 9 days. And the dosing regimen typically includes a dose on day 0, day 2, day 5, day 7, with media changes. And this is done in a serum-free culture medium. At different time points, we evaluate albumin and urea for hepatic function. We also look at ATP and GHH for cell viability. ALT, AST markers can also be looked at for clinical analysis. In the toxicity assay design, we use HEPATOPAC and stromal-only cultures. The toxicity effects observed in stromal-only cultures are subtracted so that we can identify liver-specific toxicity of the compounds and differentiate them from the general cellular toxicity of the compounds. There are different endpoints that you can utilize, such as gene expression, high content imaging, transporter assessment in this study design if you are to receive HEPATOPAC plates for evaluation internally. And as I mentioned, there are different ways to customize the assay design. The time points can be increased. The dosing can happen on a daily basis. Different time points can be selected for functional assessment. So there are different ways to go about this. I also wanted to take some time and talk about induction potential of CYP mRNA in HEPATOPAC. This Vertex study looked at the inducers, rifampin, phenobarbital, and phenytoin, and the inducers increased the expression and activity of CYP3A4, 2B6, and 2C enzymes. HEPATOPAC showed consistent and higher induction of CYP2C enzymes, which are very difficult to see in some of the standard sandwich cultures. The EC50 values of rifampin was up to 10-fold lower in HEPATOPAC than monocultures. We are currently evaluating using HEPATOPAC model for induction assessment as part of contract research services. The research is ongoing, and we are evaluating out to 14 days on how we can use some low turnover compounds and in HEPATOPAC model to evaluate their induction potential. So in summary, why use HEPATOPAC? The hepatocytes from multiple species in HEPATOPAC format are highly functional for 4+ weeks. There is longevity and better hepatocyte polarity that provides greater sensitivity. It also is very user-friendly format in standard multi-well plates, including high content imaging that can be received as ready-to-use cultures if you choose to buy HEPATOPAC as a product. For different applications that we talked about today, including the metabolic stability clearance, it is ideal for low turnover compounds because it has longer duration and single continuous incubation. This also is very helpful in MetID analysis because it allows for detection of metabolites. There are secondary or tertiary metabolites that may be formed over a few days time, and human unique metabolites, and profiling across different species in HEPATOPAC. For toxicity assays, repeat dosing regimen allows for simulating in vivo conditions and also in vivo relevant concentrations to assess toxicity effects of slowly generated metabolites, and allows users to identify what type of endpoints they want to evaluate using these models, allowing different options for functional assessment, gene expression analysis, etc. We have a lot of HEPATOPAC resources on our website. We have training videos that you can see for using the HEPATOPAC plates in your lab. We have instructions for use in different applications such as stability, MetID, toxicity. Lots of different brochures are available that walks you through study designs that you can use for HEPATOPAC. And our publication compendium has over 45 peer-reviewed publications of HEPATOPAC for different applications that are highlighted. We also have posters available on our website, which you can use this link to visit and search for HEPATOPAC, and it will show you HEPATOPAC posters. Lastly, I wanted to provide information on our upcoming webinars. The webinars we have 4 more upcoming webinars. The next one will be talking about the DILI assessment using HEPATOPAC. Then we have an siRNA webinar in HEPATOPAC, also a liver disease model in HEPATOPAC, and also reaction phenotyping methods for low turnover compounds. That will be our last webinar. With that, I would like to open it up for questions. Thank you.
Oh, thank you very much, Deepan, for a very interesting presentation. We have a number of questions coming in. One question that came in very early was actually asking if we have references available for HEPATOPAC, and I think you covered that in the last slide. But because we do have a number of references, a number of publications on HEPATOPAC, so that is available.
Certainly. Yeah, and I want to highlight that when you go look at that resource, the publications are highlighted for specific application. So it's very easy to identify. If you're looking for a transporter application, you can easily find the publications that are for that application compared to, let's say, a stability application or toxicity applications. So I would really recommend everyone to go and take a look at the resources, the compendium. And if you have any issues getting any of those publications from the website, we can certainly help you get a copy of those publications.
Thank you very much. And some other questions came in. One, Armand is asking, will the slides be shared after the webinar? And yes, they will. We will send that within a week, and if you don't get them for some reason, just reach out to us and we'll be happy to send them to you. Another question Sumi is asking, what is the rationale you would have to choose between single or pooled donors for hepatocytes, for HEPATOPAC?
For most of our stability and MetID experiments, most customers choose pool donor hepatocytes. The pool donor hepatocytes we use are 10 donor mixed gender pool, generally to basically that covers different enzyme expression profiles for certain variety of metabolizing enzymes. So it's usually the main choice for human HEPATOPAC. Sometimes certain study requires a single donor if they are looking at very specific type of application. Single donors can also be used.
It's lots of questions popping in right now. Nurad is asking, can BioAVT ship to India after that day 5 stabilization within a couple of days? And what help is available to our colleagues in India?
Currently, we are able to ship to Europe. I think we may have shipped something in Asia, but I have to look into that. I have to talk to my production team to see if they have any orders coming in from far away. I think up to 2 days. The boxes are not temperature controlled, but they are packed in such a way that the temperature is more room temperature for up to 2 days for the HEPATOPAC plates. Anything longer than that, it's not suitable for the shipment. So, but that's a good question. I would certainly inquire with our products team to see if we have ability to ship to India.
Okay, thank you.
So please reach out to us through our HEPATOPAC products page as an inquiry. Send an inquiry so somebody will reach out to you.
Sounds good. A couple of questions about the technical details. How many islands of hepatocytes will form per well? Lakshmi is asking that question.
So in the 96 well, of course, there are less islands. I would like to say maybe 8 to 10 islands in a 96 well, but I may be corrected with our products team. But usually in 96 well, there are about 3,000 to 3,200 hepatocytes. So it's not a lot of hepatocytes. With HEPATOPAC, it's all about quality over quantity. When we have used HEPATOPAC model and compared with other in vitro models, the per cell production of albumin and urea has been always higher, even when more than 10 times hepatocytes are added. So in a 96 well plate, I would say we have about 14 islands, and in 24 well format, we have about 94 islands of hepatocytes.
Oh, perfect.
I hope that helps.
And I think you answered another question too, is the number of hepatocytes countable on each island? Another comment doing is thanking you. It's saying really nice presentation and asking if you've looked at CYP2C19 induction using HEPATOPAC.
We have internally, but we are still evaluating multiple donors. We have seen some induction, but the data is not yet available for presentation. The paper that I showed, they did not look at 2C19. They looked at 2C8 and 2C9. But I think there is another publication that may have looked at 2C19. I may have to dig that up.
All right, thank you. Another question, Sylvia, thank you very much for the presentation. With a couple of questions. Are HEPATOPAC provided with a characterization panel?
What type of characterization?
I don't know, just any sort of characterization. A secondary question there. Go ahead.
Yes, so for our HEPATOPAC species that uses cryopreserved hepatocytes, we have technical sheets available. Let's say the cryopreserved hepatocytes, HEPATOPAC that we produce are using human, a rat, some mouse HEPATOPACs, and also monkey. But with dog hepatocyte and several mouse species hepatocytes, we tend to use freshly isolated hepatocytes. And sometimes the characterization information is not yet available. But for cryopreserved hepatocytes that we use in HEPATOPAC manufacturing, we certainly have technical sheets available that characterizes those cryopreserved lots. For
Yeah, I think
metabolic function.
The technical sheets, somebody's asking about the certificate of analysis.
Correct.
So if we have those available.
Yes, we certainly make them available, and we also, when we are working with the customer, we also give them options of different donors that they may choose from the donors that we have used for HEPATOPAC production. So they do have a choice in the matter in this case. So that allows them to choose a donor lot that might work better for them. And the COAs, the bio-IVT hepatocyte COAs, are extensive and available online upon request.
Thank you. Do you have any experience with HEPATOPAC using ASOs, the antisense oligos?
We have done some work on that. We have actually done some work on this, and we have some data that we have presented before. If this person would like to reach out to us directly, we can certainly share some data with them.
All right, thank you. Seeing if anybody has any additional questions popping in. I think we've answered most of the questions. One question that came in earlier: how long after receiving HEPATOPAC can studies be initiated?
The HEPATOPAC is typically received on day 6. They are shipped on day 5 for overnight shipment. Once you receive the HEPATOPAC, you have to you change media and you incubate them to stabilize them for another 48 hours. So day 8 is typically the earliest we use for incubations of standard compounds. In some use cases where we use siRNA, GalNAc conjugated siRNA or free uptake, we can start the incubations during the plating of hepatocytes, but that typically involves that typically is part of service project, but it also can be used in products as well.
Okay, I think we've answered all the questions. I will say thank you to everybody who joined us today. If you have additional questions, feel free to send them to us. You can email or reach out, and we'd be very happy to answer those offline after the webinar. So once again, thank you very much, Deepan.
Thank you. Thank you, everyone.
And we look forward to the additional webinars. We will have these every other week. You can see the next one is on September 25th. So I hope everybody can join us then.